Hello students and subscribers. This video is going to go over the light-independent reactions, sometimes called the Calvin cycle. So we've already gone through all the steps we needed for our light-dependent reactions, and so now we're going into our light-independent reactions. So a little bit of a reminder for where this occurs. I'm going to make a quick sketch of a chloroplast. Remember that chloroplasts have a double membrane, and inside the chloroplasts are grana, which are stacks of thylakoids. Woo! First one was definitely better than the second, but you get the idea. This is where the light-dependent reactions have taken place. But now we're in the liquidy area outside of those grana, and this is where our light-independent reactions are going to take place. This, as a reminder, is called the stroma. And a big key that really helps with these light-independent reactions is remembering that they are just a cycle. This refresh arrow is going to represent that we have a cycle taking place here. So to start off this cycle, we need carbon dioxide, and we need another molecule that is conveniently already in the stroma, and we abbreviate it RUBP. That stands for ribulose bisphosphate. This carbon dioxide molecule has one carbon, the RUBP has 5 carbons. So when these come together at an enzyme called rubisco, they're able to form a 6-carbon sugar. And the reason why we say 6 carbons is because we get 1 from the carbon and carbon dioxide, and we get 5 carbons from the RUBP. However, while this is wonderful that we have our 6-carbon sugar, this is only going to happen if we have an enzyme called rubisco. To remind you guys of an enzyme sketch, I'll draw a quick one here. An enzyme has an active site where substrates can fit in, and for labeling this, we'll say that this enzyme is rubisco. Here is our carbon dioxide, and here is our RUBP. When there's enough motion on this enzyme, and if both carbon dioxide and RUBP are in here at the active site, having enough motion will close up this enzyme, and so you get one product. That is our 6-carbon sugar. So I put a little star here on the cycle. It connects to the star over here, and this is just our little marginal note. Let's continue along our cycle. After being formed, this 6-carbon sugar is going to be split in 2. It's going to separate into 2 3-carbon sugars. To make this happen, we needed a pretty powerful molecule. Remember that we get ATP from our light-dependent reactions, One of those ATPs is going to get used to break up the 6-carbon sugar into 2 3-carbon sugars. So it loses that phosphate because it releases the energy held between the phosphate-phosphate bond. That energy that is released is used to break the 6-carbon sugar literally in half, because now we have a 3-carbon sugar and another 3-carbon sugar. So 3 carbons plus 3 carbons equals 6 carbons. And now that ATP has become ADP. The ATP is going to be sent back to the thylakoids, where it's going to sit very close to the ATP synthase and wait for that hydrogen ion concentration gradient. And when enough hydrogens come through and we get enough rotational energy, remember we shove a phosphate onto that ADP. Remember, we said the Calvin cycle was greedy. So the Calvin cycle takes the ATP produced by the light-dependent reactions and sends back ADP. What happens next is a series of reactions. Luckily, we don't have to worry about the names of these for now. But what eventually comes out of the Calvin cycle is a new sugar, pGal, and with a little bit more modification, pGal can form glucose. For this to happen, we use a molecule of the NADPH from the light-dependent reactions, and we steal its electron, which means we also take its hydrogen, and then we're left again with NADP+. This NADP+ is then sent back to the thylakoid. So just like the ATP came from light-dependent reactions, so did the NADPH. NADPH is utilized in the Calvin cycle, gives up an electron in those hydrogen ions, and becomes NADP+, which is just sent back so that the greedy Calvin cycle can get more NADPH. The only other thing I want to bring to your attention, the RUBP is recycled. And because we can keep using that over and over and over again, we can call this whole thing a cycle. So our light-independent reactions allows for carbon dioxide and RUBP to combine using a Rubisco enzyme to form a 6-carbon sugar. We use an ATP molecule to break that carbon sugar into 2 smaller 3-carbon sugars. Then with a bunch of processing and with an NADPH losing its electrons and hydrogens, it will become NADP+, which is also sent back to the light-dependent reactions. And as a result of going around this cycle, you get that pGal molecule, which, once modified, becomes glucose, which is C6H12O6, a very important product of photosynthesis so that plants and photosynthesizing organisms can feed themselves.